It's a pleasure to be here to present to you Pfenex and our biosimilars efforts. I'm Hubert Chen, I'm the Chief Medical Officer, and here is the first slide, which is a corporate overview of Pfenex. We are a biotech company that's based in San Diego, California. We were initially founded as a platform technology company with expertise in protein production and analysis. However, in about 2009, we decided to repurpose this platform to go into therapeutics production with a focus primarily on the development and commercialization of biosimilars. Now, our lead product in the biosimilars portfolio is a PF582, which is a biosimilar candidate to Lucentis. And for this audience, I obviously will not be going through the mechanism of action or the intended patient population for PF582. What I'd like to do, though, is to highlight some of the news that we released earlier today as part of our earnings call, in which we disclosed the top-line clinical results from our previously completed phase 1/2 study comparing Lucentis versus PF582. And we are pleased that we were able to achieve the primary objective of the study, which was to show that the safety and tolerability profiles of the two products were comparable. We also looked at efficacy in terms of visual acuity and also some pharmacodynamic parameters. Again, we were very pleased to see that there was consistent pharmacology that was demonstrated between PF582 and Lucentis. Now, as part of the announcement earlier today, we also are in the process of regaining all the rights to PF582 from our partner. So combined with the encouraging phase 1/2 study results, along with the what we believe to be a readiness for manufacturing tech transfer, we are very excited in terms of exploring all strategic options in terms of what to do with PF582 as it returns back to us, including exploring the potential for repartnering. So really that's, for the rest of the presentation though, I want to perhaps share with you Pfenex's perspectives in terms of why focus on biosimilars and why we believe that this field really will be taking off in the imminent future. And I think a good way to think about this field is to compare and contrast this to the small molecule generics, in which you can see that the latest statistics show that 80%, and this is increasing, 80% of the prescriptions that are filled in the United States are the generics. In contrast, though, as most of you are aware, the first biosimilar really was not approved by the FDA until 2015, and despite the headlines that are being generated, it's still lagging significantly behind. So really the question that we are posing is, why is there such a large discrepancy? And of course, I think most of us have seen this cartoon in terms of one of the key factors, of course, is the fact that small molecules are relatively easy to manufacture, and this is shown by the cartoon on the left-hand side in terms of the atomic structure of aspirin. On the right-hand side is your fairly typical biologic, which is a monoclonal antibody. And again, the figure really does impart to you that the complexity involved in terms of recreating a biologic really should be a lot more complicated. However, unless you have spent some time in a chemistry or a biology lab, I think we may understand this intellectually, but in terms of what does this mean, what are the practical implications, sometimes a little bit hard to grasp. So what if we swap in pictures of Lego cars? So here is a 25-piece Lego car, and if we just take a look at this picture, I think most of us feel confident that even without the assembly instructions, we can probably recreate this toy car. But what if we now swap in this Ford Explorer that was taken at Legoland in San Diego, my hometown? So now we probably start to get a little bit challenged in terms of, can I really recreate this Lego car? Well, what if, and we're all in the medical field or exposed to it, so what if we start to provide some tools so that we can use an X-ray machine from the dentist's office, we can use a Doppler ultrasound to look at the 3D structure, and we can perhaps even do a laser measuring device so we can actually figure out where to place that Ford decal to the exact millimeter. So really now, if we kind of take this analogy back to the biosimilars field, This is exactly what's happening. The chemistry, the advancements, the technology of analytical chemistry is advancing to such an extent that we're able to use mass spec, CD, and SDS page, again, to really refine and to make sure that we're actually recreating a faithful copy of the originator. So I think that's probably what I want to impart to you in terms of why the biosimilars we believe is really taking off, and Pfenex is really delighted to be a part of this effort, and we look forward to updating you on PF582 at an upcoming event. Thank you very much.